Well, good morning, guys. I'm in Proctored Keystones today, so I'm not able to be there in the classroom with you, but I did want to make sure that you are charging ahead with your genetics unit of content so that we are fully prepared for Keystones next week. So just to briefly recap yesterday, we talked about a few things. We set some of our terminology straight, and you can look back into that shared Google slide if you're curious as to what we discussed yesterday, if you need a refresher. That terminology should all be there. I haven't looked at it, but it was a collaborative project you guys were to be working on. I assume that you were keeping up with all the terminology yesterday. So things like heterozygous versus homozygous, purebred, dominance versus recessive, alleles, genes. Those are the kind of terms that you need to know before you can understand how to actually approach Punnett squares and word problems in biology. And those are important words for helping you be successful on the genetic section of the Keystones. So based off of yesterday's lesson, I'd like to kind of take things a little bit further in showing you how to pick apart a word problem and set up a couple different types of Punnett squares and then calculate frequencies based on the results of those Punnett squares. So the first type of cross that we're going to look at is a monohybrid cross, and if you know anything about me, you know that I like root words. And if you look at this term, you should see a couple of root words in it. The first being that root word mono, meaning one, and then hybrid, meaning we're mixing something up, and we're crossing two individuals. So monohybrid cross, what we do with the monohybrid cross is we take a look at how the alleles for just one trait are mixed or shuffled, and we determine probabilities or probable outcomes for a single trait. So in a monohybrid cross, we would set up a simple 2x2 Punnett square. And I call this simple because this is the simplest form of cross. A Mendelian monohybrid cross is the simplest form of genetic cross we can actually examine in genetics. Okay, so here is an example question. In the pea plants studied in Gregor Mendel's garden, round pea shape was found to be dominant to wrinkled pea shape. And then the question asks, what genotypic and phenotypic frequencies would be probable if a pea plant that is heterozygous for pea shape was crossed with a pea plant that is recessive for pea shape? And you'll notice that I didn't give you anything specifically in that question. What I didn't specifically give you were the genotypes for the plants. Instead, I gave you those terms that are going to be important for you to understand. And those terms were heterozygous, which tells us that the first set of alleles are going to look like this. They are different, meaning we have a dominant allele and a recessive allele for the gene in parent 1, and the dominant phenotype is what's going to show through in that individual. So that parent is actually going to have round peas. Okay, and so what we do is we take our dominant allele of the genotype and we put it here at the top of the table, and then we take our recessive allele and we put it over top of the second column of the Punnett square. And the X simply stands for the word cross in our term up here, monohybrid cross. So we're just looking at seed shape. That's our single trait, and we are crossing two parents to see what probabilities we could come up with for offspring. Now, the second piece of information that was given to you in this word problem is that the second parent is exhibiting the recessive trait for pea shape. And that should tell you something immediately about the genotype for that parent. That tells us that it has two lowercase alleles, two recessive alleles for that trait. So we can go ahead and distribute those in a likewise fashion down the side of the Punnett square in the bottom right-hand corner here. Okay, now the next step is to work the Punnett square out. And again, this is a simple Punnett square, 2x2. We bring our top value down, so that would be our dominant allele down, and then this recessive allele over, They join in square number 1, and then we're going to do the same thing for all of the remaining squares in the Punnett square so that we have something that looks like this. Now that alone does not answer the questions that are being asked. The actual questions, if we go back to the word problem, are asking for the genotypic and phenotypic frequencies that are probable for this cross. And so we actually have to take this information that we've generated in our table, in our Punnett square, and we have to do some maths on it, okay? So what we first have to do is we have to figure out all of the different genotypes that were created by the table. And in a quick glance, you can see here that there are only 2 different genotypes. The first being a heterozygous genotype, and the second being the homozygous, or the homozygous recessive, or just the recessive genotype. Okay, and if we count those, right, if we count those, we can see that we have 2 of each out of 4 possibilities. And so the heterozygous genotype is going to be 50% probable, the recessive genotype 50% probable, and the homozygous dominant, you can see that I included that. That is 0% probable because it didn't appear in our Punnett square up here. Now I included all 3 genotypes here because these are the only 3 in a very simple Mendelian monohybrid cross. These are the only 3 that could possibly occur. These are the only 3 that could pop up: the homozygous dominant state, the heterozygous state, and the recessive state. So we came up with our genotypic frequencies right here, 50% and 50% for heterozygous and for recessive. And what we need to do now is we need to take all of this information and we need to turn it into the phenotypic frequencies possible. And so for a round seed, we actually have to take the homozygous dominant frequency and add it to the heterozygous frequency, which in this case is 0 out of 4 plus 2 out of 4, which equals 2 out of 4, and that remains 50%. So we have a 50% chance in this cross of creating offspring with round seeds. We also have that remaining 50% chance to create wrinkled seeds, which is that recessive expression of the gene. So let's take a quick look here. What if we change the word problem around a little bit and we actually cross 2 heterozygotes for P shape? Okay, so big R, little r, big R, little r. We have a carrier for the recessive trait in both parents, but they're still exhibiting that dominant characteristic. So we're going to take these 2 heterozygote parents and we're going to cross them, and let's see how that changes our table, our Punnett square results a little bit. Okay, so you can see now there's a little bit of a different distribution of dominant to recessive alleles in the table. And that is actually going to play into our frequencies in genotype and phenotype as well. So let's have a look down here. There's the homozygous dominant genotype. We now have 1 out of 4 squares with that one in it. So 25% probability that the offspring will end up as purebred dominant, meaning all-round seeds. There is a 50% chance the upper right and lower left-hand corners of the Punnett square, 2 out of 4, 50% chance that those offspring will end up heterozygous, meaning they're carrying the recessive allele but not expressing it, and then that remaining 25%, 25%, 1 out of 4 on the Punnett square here, we're gonna take a chance at them being recessive in their genotype and also exhibiting the recessive phenotype to go with that genotype. Okay, so now our math down here has changed a little bit. So what we're going to do is we're going to have a look at how we add our homozygous dominant and our heterozygous genotypes together to get round seeds with a 75% chance due to the change in the cross that we're using. So a 75% chance now that our seeds will be round, and the remaining 25% chance that they will be that recessive, expressed as that recessive trait, wrinkled seeds. So at this point, if you need to pause and go back, feel free to do so. I'm going to move on into a little more complicated problem here. We'll call these complex Mendelian genetics. And the only one that we're really going to take a look at is the dihybrid cross. And again, if we break this down into root words, di meaning two, a cross between two individuals, di meaning we're looking at two traits, alleles for two traits, and we're mixing or creating a hybrid of those traits from two parents. Okay, so dihybrid cross, we examine the mixing of alleles for two different traits, and we determine the outcomes, the probable outcomes for the combinations of two different traits. So what that means is not only would we be looking at maybe blonde hair, but we would be looking at the probabilities of an individual receiving blonde hair with blue eyes. So a combination of two different traits, or red hair with brown eyes, okay? So the Punnett square is actually going to get a little more complicated. It's going to become a 4x4 Punnett square for dihybrid cross problems. And here's an example question. It's gonna kind of refer back to the initial question that we worked through, dealing with the shape of peas, but it's also this time going to be taking a look at the color of the pea pod. So here is the question. In the pea plant studied in Gregor Mendel's garden, round pea shape was found to be dominant to wrinkled pea shape. You already know that. While yellow pods were found to be dominant to green pods. That is new information for you. The question is asking what genotypic and phenotypic frequencies would be probable if a pea plant heterozygous for pea shape with a green pod was crossed with a pea plant recessive for pea shape and heterozygous for pod color. So let's look at our cross. Our first individual, the first parent in the cross, is heterozygous for pea shapes. That means big R, little r, and it has a green pod, which means it is going to be recessive for pod color, little y, little y. We take that individual and we cross it with a plant that is recessive for pea shape, meaning big little r, so it's wrinkled, and it is heterozygous for pod color, so we're going to actually see the big Y, little y yellow pod color expressed here. And as you can see, our table has gotten a little larger. Now things get a little bit crazy here. How did I come up with these different combinations of alleles? Well, it's almost kind of like your FOIL order of operations in math. What we're going to do is we're going to take, as soon as this little menu clears here, we're going to take the big R from the first parent and we're going to cross it first with the big Y from the second parent. Okay, so take a look there. Big R from the first parent, cross it with the big Y from the second parent, and that's how we get our first combination of alleles. And then we're going to take that same big R from the first parent and we're going to cross it with the second Y of the second parent to get big R, little y. And then we're going to take the R's from the second parent and we're going to cross them with the Y's from the first parent. So little r, and we're going to take the second R from the first parent, we're going to cross it with the Y's from the second parent. So little r, big Y, let's let that menu clear there again. Little r, big Y, that's the second R of the first parent with the Y's of the second parent. And then little r, little y for our last allele combination. And now we have to fill things in down the side, and the way that we do that is we take the R's from the second parent and we cross them with the Y's of the first parent. And if we take a quick look, we can see that the R's of the second parent are both recessive, the Y's of the first parent are both recessive, and so we can very easily come up with what our allele combinations are going to be down the side. Okay, and then it's just like the 2x2 monohybrid cross table. You bring the big R down, the little r over, the big Y down, and the little y over, okay, in the first square, which would give us a heterozygous individual for both traits. And if we extend that rule out, so I've gone ahead and filled in all of the R's for you in the table, okay, sometimes it's easier to do the first trait first and then go back and fill in the information for the second trait. Here is the table extended out, and I would ask you guys, go ahead and check that, make sure that all of my information is correct there. I do make mistakes sometimes in these Punnett squares, especially those for dihybrid crosses. So this is what our table looks like. And now, again, what we need to do is we need to actually answer the questions that were asked in the word problem. So again, it's asking for genotypic and phenotypic frequencies. So let's take a look at the math behind the genetics. So genotypic frequencies, what we have to do is we have to very carefully count the information that's in the table. Okay? So if we look, we see very first in the upper left-hand corner, we have a big R, little r, big Y, little y. We've created the heterozygous probability for both traits here. And so what we have to do is we have to go through the table and we have to count how many occurrences of this dihybrid genotype occur. So here's 1, 2, 3, and 4 out of 16 squares, which equates to 25%. The next genotype is going to be big R, little y, little y. So heterozygous for shape, recessive for color. If we count again, 4 out of 16. And then the math gets a little more complicated. Let's look at the recessive for shape, heterozygous for color. We count only 2 of those in the table, which equates to 12.5%. And then our last genotype for shape and color is recessive for both traits, little r, little y, little y. And we have 6 out of 16 of those, so 37.5%. And we need to convert those now into genetic expressions or phenotypic frequencies. So if a seed is going to be both round and yellow, it has a 25% chance of being both round and yellow, a 25% chance of being both round and green, so it is dominant for shape, recessive for color, 25% chance there. Wrinkled in yellow, recessive for shape, heterozygous for color, that is a 12.5% chance, and then wrinkled and green, we have a 37.5% chance. And these can get way more complicated depending on how we shuffle the, depending on how or what the genotypes of the parent plants would look like. So what I'd like you to do now is in your notes, let's go ahead and try a little bit of a shuffled dihybrid cross. Okay, so you're going to do a dihybrid cross for 2 plants, and both of those plants are heterozygous for both traits. So we're still talking shape represented by the letter R and color represented by the letter Y. I want you to cross 2 parents that are homozygous for both traits, and I want to see what you come up with for your genotypic and phenotypic probabilities. You can do that right in your notes. You can do it in a Google Doc, you can do it in a Google Drawing, whichever you prefer. And I will just give you a little hint. If you're not doing it in Google Sheets, if you're in fact doing it in Google Slides or Google Docs, you're going to have to use the insert table and then pick a 5 by 5 for your table size to actually drop a workable Punnett square into your doc or drawing. Okay, and then last, I am going to push some dihybrid cross problems out to the classroom stream, so please make sure that you find those and be working on those. We will be discussing them in class tomorrow. See you guys.